Hello, my name is Emily Rocha, and I am a faculty member within the neurology department at the University of Pittsburgh. And it is with great pleasure to be back at the Neuroregeneration Research Institute at McLean Hospital, where I completed my first postdoctoral fellowship. I would like to thank Penny Hallett, Will Isaacson, and Oliver Cooper for giving me this opportunity to share some of my more recent work that I've done since leaving Will Isaacson's lab. So, let me see here. There we go. Okay, so the title of my talk today is LRRK2 Inhibition Prevents Endolysosomal Deficits in Parkinson's Disease. Now, this work is really an extension of the work that I did while I was a postdoctoral fellow with Will Isaacson and Penny Hallett. And it really, these three papers that we published together as a group really form the basis of what I'm doing now. This first paper that we published highlighted that glucocerebrosidase activity, which is a lysosomal hydrolase, is reduced in normal aging as well as in Parkinson's disease patients. When we looked at preclinical animal models of Parkinson's disease, focusing again on glucocerebrosidase, we showed that glucocerebrosidase is changes in glucocerebrosidase, I should say, will affect alpha-synuclein levels. Specifically, if you inhibit glucocerebrosidase using a pharmacological agent, we were actually able to induce insoluble alpha-synuclein aggregates within dopaminergic neurons of the substantia nigra, pars compacta. And if we overexpress glucocerebrosidase using an AAV approach, we were actually able to reduce and prevent alpha-synuclein aggregates from accumulating using the Thy1 alpha-synuclein transgenic mice, which is a model of alpha-synucleinopathy. So this data really kind of led us to one of two conclusions. One, that glucocerebrosidase is the main culprit in synucleinopathies and Parkinson's, or it's possible that changes or deficits in glucocerebrosidase really actually represents something a lot larger, like overall and global lysosomal dysfunction that might be occurring during the normal aging process, and that might actually eventually lead to idiopathic Parkinson's disease if you pass, let's say, an arbitrary threshold for lysosomal function. So to study this more closely, I've been focusing on one of two pathways, the endocytic pathway or the autophagy pathway, I should say. And when you're looking at these two pathways, what you're really looking at is, for the endocytic pathway, is material that's being brought from outside of the cell to the lysosome for degradation, whereas autophagy is material from within the cell that needs to be degraded, that again is brought to the lysosome. So the endocytic pathway really focuses on material that's been internalized and surrounded by an area of cell membrane, which can then bud off inside the cell to form a vesicle containing this material that needs to be digested. And to look at the first vesicle that forms in the endocytic pathway, it's actually the early endosome. And you can use different markers to look at it, like EEA1. I chose to use RAB5 because it is specific to the early endosomes. And as you'll see later in my talk, it's actually quite important when looking at LRRK2 kinase activity. So the endocytic pathway, it goes through there's a maturation process from the early endosome to the late endosome. And what happens is you have this conversion where Rab5 will leave the vesicle when it matures into a late endosome, and Rab7 will accumulate on the outer membrane. And then eventually this late endosome can fuse with the lysosome, or in fact actually become a lysosome. So the first thing we were curious at looking at is whether or not there are changes in the early endosomes in Parkinson's disease. So we looked at postmortem Parkinson's disease patients, as well as control patients. And what we found is that Rab5 accumulates significantly within the dopaminergic neurons of idiopathic Parkinson's disease patients in comparison to control patients. And if you look at the quantification where we looked at the number of those Rab5 puncta within the dopaminergic neurons, you can see that it was significantly elevated in the Parkinson's disease patients. But not only did we find that the number of Rab5 positive puncta were up, but also the size of each one of those puncta were quite large in the idiopathic PD patients in comparison to control brains. So to study the significance of this finding, we went to the preclinical models of Parkinson's, and we like using the rotenone model for Parkinson's disease, and you'll see why in a few minutes. But there's a number of reasons why we actually enjoy using the rotenone model. But during my postdoc with Timothy Greenemeyer, we set out to kind of manipulate the rotenone dosing paradigm to try and come up with a pre-symptomatic form of the model, a pre-symptomatic model, as well as an end-stage model of the disease. And what that really meant was we wanted to find a point where the dopaminergic neurons were sick but yet not yet dying, and then also a point where we did in fact see dopaminergic cell loss. So in our hands, what we found is that if we dosed rats with rotenone for 5 consecutive days, we did not see any dopaminergic cell loss, as you can see here, and by these stereological counts of the dopaminergic neurons within the substantia nigra. But if we continued the dosing paradigm to the point where our rats were dosed about, on average, 10 days, what we saw was about a 50% cell loss of the dopaminergic neurons within the SNC. And up here are just the photomicrograph representations of what we could see in each of those regions. And when we looked at the striatum, what we found is that what we're calling an endpoint rotenone protocol caused a really nice dorsolateral lesion in the striatum, but that 5-day dosing paradigm did not cause any changes within the striatum. So if we looked at these two models using rotenone, what we found is that Rab5 accumulated quite significantly even in that acute model. So even with those 5 doses of rotenone, we saw a dramatic accumulation of Rab5. And again, this remained stable when we took our animals out to endpoint, where we saw that 50% cell loss of dopaminergic neurons. So what we were quite comfortable concluding is that Rab5 accumulates within the dopaminergic neurons of Parkinson's disease patients, as well as rats dosed with rotenone, either that acute or that endpoint protocol. So the next thing we wanted to do was look at further on in that pathway. So now that we could say that the early endosomes were definitely accumulating, well, what did the late endosomes and the lysosomes look like? So to do that in our Parkinson's disease patient tissue, we looked at both LAMP1 as well as M6PR. Because if a puncta was positive for both M6PR as well as LAMP1, it represents a late endosome. Whereas if it's positive for LAMP1 but negative for M6PR, what we were looking at more often than not is just a lysosome on its own. But you can see based on these representative photomicrographs that LAMP1 as well as M6PR were significantly reduced in our Parkinson's disease patients within those dopaminergic neurons. in comparison to control. And this was reflected quite obviously when we did our counts. So if you look at the number of LAMP1 positive and M6PR positive puncta, it's significantly down in the idiopathic PD patient tissue. And this was also true for our lysosomes, so that LAMP1 positive but M6PR negative puncta. And next, we wanted to know if we were able to replicate this finding in our preclinical animal models using both that acute rotenone dosing paradigm or that endpoint rotenone dosing paradigm. And you can see quite obviously that the rotenone, even 5 doses of rotenone, caused a reduction in the number of late endosomes. And this was also true when we took our animals out for that 10 to 14 doses. And if you looked at the number of lysosomes, you can see again that the number of lysosomes was down in our acute as well as our endpoint stage animals. So now that we were quite comfortable saying that there's really something, this whole endocytic pathway is significantly being affected, well, we wanted to know what was going on within the lysosomes. So we chose to look at glucocerebrosidase, which is up here short formed as GCase. And you can see that GCase was significantly down in our postmortem Parkinson's disease patient tissue, again, as well as LAMP1. And this was reflected in our analysis when we looked at the number of GBA positive and LAMP1 positive puncta within the TH neurons, significantly down in our idiopathic PD patients. And when we looked at rats, we looked at a couple of different measures when looking at GCase activity. So at first, we looked at GCase activity itself as a functional measurement of lysosomal function. And you can see we saw a 25% reduction in GCase activity in rats that received only 5 doses of rotenone, so that acute paradigm. And this also was true if we took our animals out to endpoint, which to me I thought was quite significant because at this point we actually have a 50% cell loss. So these are looking at the animals that are, the cells that are surviving within the substantia nigra. And next we also looked at glycosylceramide in both our acute and our endpoint models. And glycosylceramide really didn't accumulate in the acute paradigm in any of the brain regions that we looked at, so the striatum, the cortex, or the hippocampus. But if we took the animals out longer to their endpoint, what we saw was a significant accumulation within the hippocampus, and we saw a trend towards significance within the striatum, but it wasn't quite significant. And again, we didn't see anything in the cortex, which that wasn't really all that surprising to us because we've seen that before where we don't really see a change in the cortex. And next, we decided to look at ceramide. And when looking at ceramide, again, we didn't really see much in the animals that received only 5 doses of rotenone, but the animals that were taken out to endpoint, we saw a significant reduction in the striatum, the cortex, and the hippocampus when we looked at our ceramide levels. So this suggests that there's lysosomal dysfunction occurring within both our idiopathic PD patients as well as our animal models of the disease. And to take that one step further, we chose to look at Cathepsin D, mainly because it's one of the main proteases within the lysosome that degrades alpha-synuclein. So when looking at Cathepsin D, you can see that there again is a reduction in the number of LAMP1 puncta in our idiopathic PD patients. Now, Cathepsin D surprisingly accumulated quite dramatically in our idiopathic PD patients. But if we tried to co-localize our Cathepsin D with LAMP1, what we actually saw was that there was a high number of cytosolic Cathepsin D puncta in our idiopathic PD patients. When we looked at our lysosomal Cathepsin D, we actually saw a reduction. And it's possible what we were seeing here is a permeabilization of the lysosome where things are starting to, or proteases are starting to leak out of the lysosome. When we looked at our preclinical models, we were able to replicate this same finding. So we saw an accumulation of Cathepsin D in our acute model, as you can see here in the representative image, in comparison to our vehicle. And again, when we look at endpoint, that Cathepsin D puncta is significantly elevated within the dopaminergic neurons. But when we tried to colocalize Cathepsin D with LAMP1, we saw again a significant reduction only in our animals that were taken out to endpoint. Again, suggesting that it's possible that what we're seeing is a lysosomal membrane permeabilization occurring. And all of these changes really suggest that in both idiopathic Parkinson's disease as well as our animal models, we do see lysosomal dysfunction. And with that, it wasn't surprising that in our preclinical models, we saw an accumulation of alpha-synuclein within the dopaminergic neurons, even with only 5 doses of rotenone. So what we're looking at here is endogenous accumulation of alpha-synuclein within the dopaminergic neurons. And this is one of the main reasons why I like using the rotenone model to study some of the changes that occur within Parkinson's disease. So again, we see this accumulation both after 5 doses of rotenone, which stays elevated at endpoint. Again, when we see that 50% cell loss of dopaminergic neurons. Now, several years ago, our lab published a paper looking at LRRK2 activity. Now, those of you who study LRRK2 are well aware of the difficulties in looking at LRRK2 kinase activity, especially in specific cell populations. But using the proximity ligation assay, Roberto Di Maio was able to show that we have an accumulation or an increase in LRRK2 kinase activity in the dopaminergic neurons of idiopathic PD patients, as well as an increase in phospho-Rab10 within these same neurons. And again, when looking at our preclinical models, so animals that were dosed with rotenone until they were at their endpoint, we were able to see an increase in LRRK2 kinase activity. So it's possible that increased LRRK2 kinase activity impairs lysosomal function in idiopathic Parkinson's disease, in part through excessive phosphorylation of Rab5. So it's possible that overactivation of LRRK2 kinase can actually impair the early endosomal pathway by allowing excessive phosphorylation of Rab5. So inhibition of LRRK2 may be beneficial to the greater idiopathic PD population, so not just those that harbor a LRRK2 mutation. So to look more closely at this, we decided to use a LRRK2 kinase inhibitor, so PF360. PF360 was distributed from Pfizer. So our design was to use rotenone, again, our same dose of 2.8 mg per kg IP, and we dosed our rats in the presence or absence of PF360. And PF360 was given twice daily by oral gavage. And the dosing regimen was all predetermined based on pharmacokinetic data that was shared with us from Pfizer at the time. So we ended up with 4 groups, vehicle on its own, rotenone by itself, PF360 by itself, and then the combination of both PF360 and rotenone. And the first thing we thought we should do was evaluate our target engagement. So really, does our dosing paradigm using PF360 in fact block LRRK2 kinase activity. And we chose to do this using the proximity ligation assay targeted towards LRRK2. And you can see that we have this nice increase in LRRK2 kinase activity in our rats that received rotenone. But in those that had PF360 on board with rotenone, you can see that clearly there isn't any LRRK2 kinase activity. And we also, just to further confirm that we did achieve our target engagement, we also looked at phospho-Rab10, which is a substrate of LRRK2. And you can see nice accumulation of phospho-Rab10 in our rotenone-treated animals within the dopaminergic neurons. And over here is the quantification that goes along with that. So that nice accumulation of phospho-Rab10. But when we give PF360 with rotenone, we clearly blocked the phosphorylation of Rab10, again highlighting that this dosing paradigm that we did elect to use did in fact block LRRK2 kinase. So the first thing that we wanted to do once we felt that we were quite satisfied with our dosing paradigm was to look at Rab5. So when we give rotenone, we saw that really nice accumulation of just total Rab5 within the dopaminergic neurons. And this is quantified over here. And if we put PF360 on board, we very clearly blocked that accumulation of Rab5, suggesting that that early endosome is in fact probably able to mature into the late endosome. And when looking at PF360 in our, looking at alpha-synuclein, what we did in fact find was that, again, rotenone caused that endogenous accumulation of alpha-synuclein, but PF360 was absolutely able to prevent that accumulation from occurring. And just for the sake of time, I didn't show the late endosome and the lysosomes, but PF360 did in fact allow the maturation to the late endosomes occur, and the number of lysosomes looked quite similar to that of controls. So when we looked at the degeneration, and this is just the quantification that goes along with the alpha-synuclein. So you can see that rotenone caused an accumulation of alpha-synuclein, and PF360 was in fact able to prevent that from occurring. So one of the last things we wanted to do was look at dopaminergic survival, and we did this using stereological technique within the substantia nigra. And we can see that rotenone caused about a 50% reduction in the number of Th-positive neurons, and PF360 did in fact prevent the cell loss caused by rotenone. And this also was quite evident when looking at the striatum. So we see this nice dorsolateral lesion in our rotenone-treated rats that was completely blocked in animals that received PF360 with rotenone. So this data really suggests that blocking LRRK2 might be therapeutic for idiopathic PD patients, but are LRRK2 kinase inhibitors really the best approach to treat Parkinson's disease? There's been data out there to suggest that LRRK2 kinase inhibitors can actually cause pulmonary defects. So we wanted to know, or we were curious about looking at whether or not there's an alternative approach that can target LRRK2 that would have less off-target effects. So we chose to team up with Biogen and Ionis to look at whether or not LRRK2 antisense oligonucleotides could be beneficial in comparison and not have the same off-target effects as PF360. So currently, Biogen and Ionis have LRRK2 antisense oligonucleotides in clinical trials at the moment. Its primary objective is just strictly safety and tolerability of single and multiple doses to participants with idiopathic with patients with Parkinson's disease. Their secondary objective with this particular clinical trial is to evaluate the pharmacokinetics of using antisense oligonucleotides to LRRK2. This study is actually open to PD patients with and without a LRRK2 mutation. The start date for this particular study was in 2019, and it's expected to be completed in 2022. So it'll be very interesting to see what the outcome of this particular clinical trial will be. But we decided to look at whether or not antisense oligonucleotides against LRRK2 could be as effective as PF360. So we did a study with these two head-to-head, and what we to do this, for the antisense oligonucleotide approach, we did an ICV ASO delivery. We had a 3-week incubation period before we started our endpoint rotenone dosing paradigm. So these rats received anywhere between 7 and 10 injections of rotenone. The ASO was a single bolus injection to the intracerebral ventricular system, and they received about 1,000 micrograms of LRRK2 ASO or a control. And we, in parallel, did a PF360 study, very similar to the study that I just presented, where animals received either rotenone on its own or in the presence or absence of PF360 twice daily. So the first thing we wanted to do in this particular study was really evaluate whether or not our oligonucleotide delivery system did in fact hit the entire ventricular system. So using a 0.2 methylene blue, you can see that by targeting this part of the lateral ventricle, we were able to successfully reach the entire ventricular system. And when we looked at the ASO against LRRK2 spread 3 weeks following surgery, we used an antibody that was specific to the ASO, and you can see very nice spread through multiple brain regions. So we could see the ASO in the cortex, the striatum, the hippocampus, the substantia nigra, as well as the locus coeruleus. And on this bottom row, you're looking at just a magnification in these same brain regions. So looking at 60x within the cortex, striatum, hippocampus, substantia nigra, and the locus coeruleus. So again, for target engagement purposes, the first thing we elected to do was look at the mRNA for LRRK2, because as we all know, the ASOs will target the mRNA expression of a particular protein. So the ASO against LRRK2, we saw about a 70% reduction in the cortex of rats that received the LRRK2 ASO. And when we looked at the mRNA of LRRK2 in our PF360 rats, you can see the mRNA really didn't change, which is comforting. And the when we looked at, so the first thing we wanted to look at is whether or not these two therapeutic approaches were able to prevent dopaminergic cell loss. So using a stereological approach, you can see that we saw a nice reduction in the number of dopaminergic cells in our animals that received rotenone alone in our ASO cohort, as well as in our animals that received PF360 in the PF360 arm of the study. So rotenone alone again caused a nice reduction in the number of surviving TH neurons. But in our animals right here that received the ASO against LRRK2 and rotenone, we were able to prevent dopaminergic cell loss. And that was also true when we looked at the animals that received PF360 and rotenone. So we were quite happy with our PF360 arm of the study because it really showed that we were able to replicate our previous findings. In summary, what I have shown to you guys today is that deficits in the endocytic pathway and lysosomal function can occur in idiopathic Parkinson's disease patients. And we are able to replicate these deficits using rat models of Parkinson's disease. LRRK2 kinase activity is in fact elevated in dopaminergic neurons of idiopathic PD patients. And it's possible that this increase in LRRK2 kinase activity may be responsible for the deficits that we observed in the endolysosomal pathway. And LRRK2 kinase inhibitors, as well as an ASO against LRRK2, may be viable therapeutic targets in the future to treat idiopathic PD patients, in addition to those that have a mutation. And it's possible that an ASO against LRRK2 may be a more attractive approach to target LRRK2 because it may have less off-target effects. So I'd like to just thank everybody who contributed to all of the work up to this point. Timothy Greenemeyer, as well as Terry Hastings as my mentors during my postdoc, as well as members of the Greenemeyer and Hastings lab. Julia Koffler within the neuropathology department at the University of Pittsburgh, who provided me with all of the postmortem tissue, as well as the lysosomal group at Merck at West Point, who really looked at a lot, who did the mass spec data for us, looking at glucocerebrosidase and ceramide, and then all of our funding and the different funders that helped pay for all of this work. I'd like to thank. So thank you all for listening, and please reach out if you have any questions. So thank you very much.